Now that we have investigated subcellular events in aging, we can go even deeper to the molecular level. So let's talk about protein damage first. In a sense, aging is just like driving a car. Over time, the wear and tear from normal operations causes individual components to fail and break down. In a car, this damage can be easily localized and repaired. The controlled explosions within the engine damage the metal parts containing these explosions, leading to decreased performance, leaky engines, and whatnot. Tires wear down, gears experience friction and become degraded. Mitochondria are to cells what combustion engines are to cars. They perform essentially the same chemical reaction, the oxidation of hydrocarbon fuels, and therefore get damaged by them. But this is where the analogy ends. Proper maintenance and replacements of failing parts may extend the lifespan of a car. But with a cell, this is difficult. For a car, the replacement parts are from the outside, in the hands of a mechanic. But the instructions for repair of cellular components reside within the cell, the same cell that experiences the damage. If those instructions get damaged themselves, repair becomes impossible. So far in this course, we have talked a lot about damage. Damage on an organismal level, damage to organs and tissues, damage to cells and even subcellular compartments, without really explaining what damage means. In the penultimate module of this course, we will talk about the molecular nature of this damage, particularly on the protein and DNA level. Proteins are made from 20 different amino acids, small molecules with an amino group at one end and a carboxy group at the other, connected by a carbon with a variable side chain R. To form a protein, individual amino acids are connected via peptide bonds between a carboxy group of one amino acid and the amino group of the next amino acid. In this way, a carbon backbone is formed from which the side chains R stick out. Since these side chains have different sizes and chemical properties, the interaction between these side chains thus determines how a protein folds and its overall three-dimensional shape. The shape of a protein is therefore directly determined by its primary structure. In other words, the particular order of the amino acids within the polypeptide. This primary structure thus directly determines the higher order structures of secondary, tertiary, and even quaternary structure. The 20 different proteinaceous amino acids can be roughly grouped according to the chemical nature of the side chain R. There are positive and negatively charged side chains, non-charged but polar side chains, and non-polar side chains. Protein damage, therefore, is essentially a chemical modification of these side chains, which in turn results in altered interactions of side chains within their secondary and tertiary structures. This, in turn, may lead to protein destabilization, misfolding, and/or functional degradation. Due to the difference in chemical nature of the side chains, chemical modifiers will affect these side chains in different ways. Some of the chemical modifications of amino acids are part of normal cellular reactions, shown in blue in this figure, and are required for proper regulation of protein function. These reactions are carefully controlled and executed by specialized enzymes. Other modifications simply occur because the chemistry of the water environment inside the cell is simply such that it permits certain chemical reactions. These reactions, shown in green in the figure, are not enzymatically regulated and occur stochastically. Some side chains are vulnerable to oxidation reactions, especially those containing sulfur or oxygen groups, such as cysteine, tyrosine, or serine. Amino acid side chains containing amino groups such as arginine or lysine are prime targets for reactions with carbohydrates, which then forms the advanced glycation end products, or AGEs, that we have discussed earlier in this course. Some side chains, such as asparagine and glutamine, may lose the amino group altogether in a deamidation reaction, turning from a neutral amino acid into a charged amino acid. Protein damage is thus dependent on the type of damaging agent, the type of protein, and which amino acids are prevalent within that protein. Also on where susceptible amino acids are located within the protein. If they are buried deep within the protein, they may not ever get exposed to the damaging agent and remain unmodified. If they are on the protein's surface, however, they may readily react, which may destabilize the protein, expose internal amino acids, which in turn now may react chemically, thus accelerating the protein damage. Oxidative protein damage occurs via radical formation. We already talked at length about how the electron transport chain within mitochondria can create superoxide radicals. These superoxide radicals may also be created by other cellular sources, such as NADH oxidase. Superoxide is so reactive it must immediately be converted into hydrogen peroxide by SOD. Hydrogen peroxide in turn may undergo further chemical reactions, for example disintegrating into hydroxyl radicals, which are especially good at reacting with metal ions that are in the active site of many enzymes. The hydroxyl radical may also readily react with ubiquitous halogen ions, especially bromide and chloride, to form hypochloric or hypobromic acids. Bleach, essentially. Thus, hydrogen peroxide needs to be detoxified as well, through the action of the enzymes catalase, glutathione peroxidase, and peroxyredoxin. Interestingly, these RAS are not the only oxidizing agents in the cell. Reactive nitrogen species, or RNS, may also be generated by the action of superoxide. The enzyme nitric oxide synthase creates nitric oxides, or NO, from the amino acid arginine. NO is an important signaling molecule involved in regulating vasoconstriction of blood vessels or even nerve signal transmission. This molecule is small enough that it can simply diffuse through tissues and subcellular compartments and can easily react with superoxide radicals. This reaction yields peroxynitrite. Peroxynitrite is especially good at oxidizing the side chains of the amino acids cysteine, methionine, tyrosine, and tryptophan. Moreover, peroxynitrite also reacts with the cellular carbonate buffer system, eventually creating nitrogen dioxide and carbonate radicals, which in turn can initiate a whole slew of other radical reactions. But cellular reactions are not the only source of radicals. Sunlight, for example, may directly create peroxide radicals with all the radical chemistry that entails. The most important amino acids to be damaged or modified by ROS or RNS are amino acids with highly reactive functional groups in the side chains, especially those with sulfur, nitrogen, or oxygen. Thus, cysteine, methionine, tryptophan, tyrosine, phenylalanine, and histidine are usually the most changed amino acids. For example, the thiols of cysteine can be oxidized to thiol radicals, hydroxy groups and tyrosine can be oxidized to peroxide, and especially tryptophan can easily form nitrosylation products. The offshoot of all these oxidation reactions is that protein stability is lost. Cysteine can form so-called disulfide bridges that are critical in stabilizing tertiary protein structures. Tyrosine can form hydrogen bonds, and so on. When these amino acid side chains are oxidized, these normal stabilizing interactions are lost and replaced by new, destabilizing chemical groups. As a consequence, the protein may slightly unfold, thus exposing parts of its interior towards the outside. The exposure of these normally buried sequences, also known as dagrons, triggers the cellular recycling machinery for proteins, the proteasome. The proteasome system binds dagrons, which leads to the degradation of the affected protein. Cells have sophisticated systems in place to mitigate oxidative damage. The activity of the ROS scavenger enzymes discussed earlier is tightly regulated in order to assure that ROS generation does not become too high, which may lead to too much cellular damage and functional impairment. At the same time, however, some ROS generation is required, as ROS and RNS may serve as signaling molecules and at low levels actually stimulate detoxification mechanisms. However, this means that oxidative damage cannot ever be completely abolished, as this would lead to functional impairment similar to too much ROS generation. Since damage therefore can't be avoided, additional mechanisms exist to repair already damaged proteins. Unfortunately, Due to the nature of the chemical modifications caused by RNS and ROS, most damage to amino acids is irreversible. Damage that can be repaired mostly involves the sulfur atoms in cysteine and methionine amino acids. Oxidized sulfhydryl groups in proteins react with reduced sulfhydryl groups in the molecule glutathione, thus passing on their damage to this small peptide. In essence, glutathione and damaged proteins simply exchange the damage. Glutathione inherits the damage from the protein, repairing the protein in the process. Glutathione also plays a role in damage prevention by soaking up and detoxifying radical species and helping to repair the activity of antioxidant molecules such as vitamin C. In order to regenerate glutathione, a series of redox reactions is carried out involving glutathione peroxidase and periredoxin to restore and replenish the glutathione antioxidant. The glutathione system is the most abundant cellular redox system and is thought to play a major role during aging. Interestingly, the GSH to GSSG ratio, in other words, the ratio of the reduced form of glutathione to its oxidized form, decreases with age, indicating that a more oxidized cellular environment exists at advanced age and decreases with age, indicative of a more oxidized cellular environment and reduced regenerative capabilities. This has been dubbed the redox theory of aging, another refinement of the free radical theory of aging. Proteins may also be damaged by non-oxidative means, most prominently via deamidation, the hydrolysis of the amido group in the amino acids asparagine and glutamine, which results in a net conversion of a neutral amino acid into a charged amino acid. Another important modification is carbonylation, the addition of an aldehyde group, which occurs particularly often at cysteine, lysine, and histidine amino acids. Aldehydes are highly reactive carbonyls that in turn may react with other functional groups, which may lead to protein-protein cross-linking, or even cross-linking within a single protein. Thus, carbonylation favors protein aggregation and loss of protein function. Protein carbonyls, especially 4-hydroxynonenal or H&E, accumulate with aging and in many aging-associated diseases. Reduction of H&E accumulation may even lead to increased longevity in certain model organisms. As we have seen, protein damage can take many forms. In its simplest, proteins may merely lose their native structure and denature. If this protein unfolding occurs, a specialized set of enzymes, the chaperones, refold the enzyme back into its native state in a process that requires energy. However, if a cell is energy starved, for example, due to inefficient mitochondria, or protein damage is more severe, the damaged protein must be removed from the cell and degraded. Simply excising and repairing the damaged amino acid is, unlike with DNA repair, not possible, as this would involve the destruction of the polypeptide chain. Thus, by and large, damaged proteins are degraded through the cellular recycling systems. The two major cellular recycling systems are the proteasome system and the lysosomal system. The proteasome system is largely responsible for degradation and recycling of soluble cellular proteins, while the lysosomal system is responsible for the recycling of large organelles and insoluble protein aggregates. The ubiquitin proteasome system, or UPS, regulates the amount of protein turnover in the cell, not only of damaged, but also incorrectly synthesized, as well as healthy proteins. As such, it is key for protein homeostasis and the regulation of activity for certain proteins. It consists of the proteasome, a large multi-subunit protease that feeds proteins through a barrel-like structure and breaks them down into individual amino acids. The second component is the ubiquitin system, a 3-part enzymatic cascade that results in the tagging of proteins that are to be degraded with the small peptide ubiquitin. The proteasome can recognize ubiquitin-related proteins and thus degrade them. During aging, the function of this system generally declines, but because there are many different types of proteasomes and ubiquitin systems, this process remains poorly understood. Moreover, sometimes extensive protein damage leads to the generation of insoluble protein aggregates that the UPS can no longer clear from the cell. It has even been suggested that the UPS may get clogged up by protein aggregates, leading to loss of efficiency. Regardless, the consequences of loss of UPS function are severe, as damaged proteins can no longer be recycled and protein quality control is no longer performed adequately. This leads to the accumulation of non-functional enzymes and the general decline in cellular functionality. This can be clearly demonstrated by administration of proteasome inhibitors to healthy cells. These cells quickly enter a senescent phenotype. Conversely, senescent cells can be restored via reactivating the UPS. Longevity in worms can be increased by overexpressing components of the UPS. In the same vein, fibroblasts from centenarians generally have higher proteasome activity, further highlighting the importance of protein quality control for health and longevity. The other major proteolytic system is the lysosomal autophagy system, which is designed to take care of larger particles, including organelles, pathogens, or even large insoluble protein aggregates. Autophagy, or cellular self-digestion, begins with the assembly of an autophagosome. This step involves the interplay of a plethora of cellular proteins that work together to build a membrane de novo around the particle to be digested. Once the autophagosome has been created, it can then fuse with a lysosome, thus exposing its content to the lysosomal digestive enzymes, including proteases, lipases, nucleotidases, and glycosidases. Together with a low pH in this organelle, this set of enzymes ensures that the cargo that arrives at the lysosome is degraded into its individual building blocks, which then can be recycled. Just as the UPS, autophagy declines with age. The reasons and consequences for this are similar. As more and more damaged cargo gets delivered to the lysosome, efficiency of the digestive enzymes decreases. A variety of reasons account for this. Sometimes damaged proteins or lipids form insoluble aggregates, which are difficult to attack by these enzymes. As a consequence, these aggregates remain in the lysosome for far longer than normal, which leads to a rise in pH and a further decline in degradative capability. In addition, the lysosomal membrane begins malfunctioning, which could lead to leakage of lysosomal enzymes out of the lysosome, which decreases lysosome function. Lysosomal membranes also begin to fail in fusion efficiency, which results in the persistence of cytosolic autophagosomes as well as reduced delivery of digestive enzymes to the lysosome. Moreover, one of the end products of lysosomal activity is lipofuscin, often called the age pigment. Lipofuscin accumulates with age and may be used as a biomarker of aging. Due to its insolubility, however, it accumulates within lysosomes and leads to increased functional decline of this organelle. And just like the UPS, inhibiting autophagy induces cellular senescence, while blocking autophagy in C. elegans shortens their lifespan, further highlighting the importance of these cellular quality control systems to aging and longevity. Check your understanding. Define different levels of protein structure. Explain different types of protein damage and their effect on protein structure and function. Describe the different types of cellular protein quality control systems. Explain the connection between cellular quality control and aging.